Okay, everyone, so in our last lecture, we did respiratory anatomy from the nares, or the nostrils, the openings into the nose, through the nasal turbinates, and around through the sinuses, maxillary, ethmoid, sphenoid, and frontal, where the air was warmed, humidified, and filtered. Then it moved down into the nasopharynx, oropharynx, and laryngopharynx, where the epiglottis would allow air to travel into the trachea, down the trachea with its C-shaped cartilage rings, into the two main bronchi, then secondary bronchi, smaller and smaller bronchi, until we got to the bronchioles, and then eventually the alveoli. So today we're going to talk about physiology of the respiratory system. So let's take a moment and think of what the functions of the respiratory system are. You may think this is obvious, but I challenge you to take a moment and see if you can come up with at least 3 things. All right, let's see how you did. So obviously it's the respiratory system, it's going to do respiration for us. But then the question is, what is respiration? You might say it's breathing. And sort of, yes. Respiration is actually the process by which we bring oxygen into the body, right? Because our mitochondria need it in order to do aerobic cellular respiration to make those 36 ATP from a glucose. We also need to release carbon dioxide out of the body. So carbon dioxide, you'll recall, is created as a waste product from aerobic cellular respiration, and so we need to get rid of that. Interestingly, the respiratory system also has some very important additional functions that have nothing to do with respiration. So we've already talked about how the respiratory pump assists with venous return. That's when the diaphragm moves down, creates negative pressure in the thoracic cavity, and helps pull blood through the venous system back toward the heart. Also, our respiratory system is what allows us to speak, right? And speech and communication is phenomenally important for humans. It also contributes to olfaction, or a sense of smell. And we talked about that last time, how the olfactory bulb sends nerve fibers down into the nasal cavity. It also has some important immune functions. Remember, we talked about the tonsils last time are housing immune cells. And our respiratory system is also part of how we maintain homeostasis in regard to not just oxygen and carbon dioxide, but regulation of blood pH, which is a variable that we need to control very, very tightly. Because if pH changes, then the configuration of proteins change. They change in their shape and they can't function properly. And many proteins are enzymes, and if enzymes can't work properly, then you're really in a difficult place. So what are the actual mechanics of respiration, right? So how do we move air in and out of our body, which is what allows us to bring oxygen in and to blow carbon dioxide out? So respiration is also called ventilation. Ventilation means the movement of air through a space. So you'll hear that word used. That's why we also call the machines that breathe for people when they're really sick and we have to breathe for them. That's why we call those ventilators, right? Getting air in and out. So there are 2 phases of respiration or ventilation. There's inspiration, which is not the like, oh, I'm inspired to create a masterful artwork, but it's inhalation, right? So you inspire is when you bring air into your lungs, and then expiration is what you may think of as exhalation, or pushing the air out of the lungs. So my question for you is, which phase of respiration do you think is active and requires work and therefore energy in the form of ATP? And which phase of respiration do you think is passive? So take a few breaths in and out, maybe pause the recording, and make your best guess. So it actually turns out that inspiration is the active process that requires energy. So for most of the time, when we're doing quiet inspiration, what happens is that our diaphragm over here, our diaphragm is going to contract. It's this dome-shaped muscle, and when it contracts, it moves downward. So it's kind of like the plunger on a syringe, right? So the diaphragm moves downward, pushes down into the abdomen, and that creates negative pressure inside the thoracic cavity. The external intercostal muscles, which you'll remember kind of go from up and out to down and in. They have that direction as if you're putting your hands in your pockets. Those muscles, when they contract, they kind of elevate the ribs upward and outward. We're usually only going to use those for really deep inhalation. So go ahead and try to take as big a breath in as you can. And really feel towards the end of that how you're lifting your ribs upward and outward to create more space, right, to expand the size of the thoracic cavity, therefore creating negative pressure or suction, which then is going to pull air into the system. So there are a couple of videos that I've posted in Canvas. This is a great 3D animation of what it looks like as the diaphragm is contracting. So in order to allow for this to work, we need to make sure that the air that gets pulled in is going to enter into the lungs and that the lungs are going to expand and fill with that air. We also need to make sure that as the lungs expand, as air moves into them, that there's no friction between the outside of the lung and the inside of the rib cage. It'd be really uncomfortable if you had this, rough rubbing all of the time. So you probably remember from the beginning of the semester that the thoracic cavity, the pleural cavity, is lined by pleura, right? So we had pericardium surrounding the heart and pleura surrounding the lungs. So this outer layer here, which is attached to the inside of the rib cage, is the parietal pleura. And then this inner layer, which covers the surface of the lungs themselves, is the visceral pleura. So you have these two thin, slippery membranes, and then this little tiny space in between them. That space contains a very small amount of fluid, which keeps things nice and frictionless and allows for those two membranes to glide past each other. So not only does this allow for a nice, smooth, frictionless gliding surface, it also maintains the vacuum that's necessary to make sure that the air actually gets pulled into the lung tissue itself. This is an excellent video also posted in Canvas that I'm going to ask you to watch now because they do a really great animation of what is happening in the thoracic cavity as a result of the forces in that pleural space. So if the pleura don't do that job and kind of keep the lungs stretched out and attached to the inside of the rib cage through that process called mechanical coupling, then the lung won't necessarily inflate. So this can happen if you have a hole in one of your lungs, for example, that allows air out into this pleural space. So if that happens, you lose that connection, right? That mechanical coupling of the lungs against the inside of the thoracic cavity, and air can accumulate in this space outside of the lung. This is called a pneumothorax because pneumo means air and thorax, thoracic cavity, and can be really problematic, obviously, because then your lung doesn't expand, and so you can feel really short of breath because you're not able to ventilate as well as you should. This is a chest x-ray of somebody who has a collapsed lung or a pneumothorax. So I'm just going to show you what we're seeing. First of all, you can see the ribs, you can see the clavicles, you can even see the scapula here, right? This here is the heart and the big blood vessels coming out of it, like the aorta and the pulmonary trunk. On this side over here, the patient's left side, we can see all kinds of little beebly things out in the lungs, right? It's not totally 100% black, right? You can see that there's tissue there. It's mostly filled with air, but there's tissue there. However, if we look over on the right side, look at how deep black this is. This is like deep space, right? There is nothing in there except for air. There is no lung tissue in that part of the thorax. And in fact, this right here is their lung, which is completely deflated and is probably being squished. This is probably what's called a tension pneumothorax by how much air is in that thoracic cavity. So pleura are important. So to kind of summarize what happens during inspiration, the diaphragm is going to contract and move downward, increasing the space in the thoracic cavity. So then if you remember physics and Boyle's law, that creates negative pressure like a vacuum that is going to pull air into the system. Because the pleura keep the lungs attached and maintain that vacuum, air has no place to go but into the lungs. We only use our external intercostal muscles when we're doing deep inspiration. Usually normal quiet breathing, we're just using our diaphragm. Now, what about expiration? So this is generally passive. So expiration is the passive phase, and the diaphragm relaxes. And when the diaphragm relaxes, all your organs and stuff in your belly kind of push it back upwards. And so therefore, the pressure inside the thorax decreases because now you have the diaphragm pushing up into the thoracic cavity, and so the air gets pushed out. This is a passive relaxation process. Of course, sometimes you want to exhale more forcefully, right? So if you're singing or if you're yelling or you're just blowing out really hard and fast trying to blow out some candles, or if you're sneezing, right, which is an involuntary forced exhalation, then you have some additional muscles that can help. So go ahead and take a big breath in and then try to blow out as hard and fast as you can. And then say to yourself, okay, what muscles did I just use? And if you felt some muscles in between your ribs, kind of trying to squeeze your rib cage down and in, you were right. Your internal intercostal muscles, right, that have the fiber direction going the other way, not really well shown here. It's more like this, right? Your internal intercostals are going to contract and pull the ribs down and in. You're also going to use your abdominal muscles. So remember your muscles of the abdominal wall. So to help kind of squeeze your abdominal cavity and push that diaphragm even further up into the thoracic cavity, therefore decreasing its size and pushing more air out. So we've talked about the mechanics of respiration, how we bring air into our body through the diaphragm contracting, creating negative pressure in the thoracic cavity, and how we breathe out, expiration, with the diaphragm just relaxing back. What actually controls respiration, right? Who's in charge? Of course, it's the central nervous system, and of course, it's my favorite part of the brain, the medulla oblongata. Right? So we've talked about before how the medulla oblongata is kind of like your life support center. Last week we talked about how it activates the sympathetic or parasympathetic nervous systems to regulate heart rate, cardiac output, blood pressure, right? It also has a respiratory center, which is responsible for that automatic unconscious breathing that we're doing all the time. So usually at rest, most people breathe somewhere between 6 to 12 times per minute. It depends on a lot of different variables. And the medulla oblongata is pretty smart in that it has some systems that we're going to talk about so that it can increase the respiratory rate or the number of times per minute that you breathe when needed, therefore increasing the volumes of air that you're circulating through and your ability to get oxygen and get rid of carbon dioxide. Now, the interesting thing about breathing is, unlike the heart, we can also do voluntary control, right? So we can purposefully change our rate and depth of respiration at will, right? We can have conscious control over this, but only to a point, right? So if you try to hold your breath, at a certain point, you're not going to be able to hold your breath anymore. Also, if you choose to hyperventilate, which I don't recommend, it'll make you feel lousy, but if you hyperventilate, again, you can only do that for so long before your body is going to force you to stop because you're throwing off the pH way too much, and we'll talk about that a little bit. But we do have voluntary control, which is part of what allows us to speak and sing and blow out candles and all those things. So how does the medulla oblongata know when we need to breathe? So if we were in class, I'd have you have a conversation with each other about this, but alas, so take a moment, see if you can figure out what the stimulus for the medulla oblongata might be. So most people are going to say it's because we need more oxygen. Now, for the most part, I have to tell you, this is not usually how it works. We do have some receptors in the brainstem that can sense if oxygen levels are too low. But this is really infrequently. It's really infrequent that this happens, right? When you're healthy, your oxygen levels actually almost never get too low. And the reason for that is that there's more oxygen that we need in each breath of air, right? So there's about 21% oxygen in the air that we breathe in, and the air we breathe out still has about 14% oxygen. So we're not even using all of the oxygen that we're breathing in and out, which is really useful because that's why rescue breathing can work, or mouth-to-mouth resuscitation, right? And we also know that this can't be the prime driver of how often our medulla signals the diaphragm, because if you put an oxygen mask on somebody, it doesn't actually slow down their breathing if they were in a normal non-disease state, right? So if I put an oxygen mask on you, it's not going to make you decrease your respiratory rate. Only in the presence of lung disease, so problems like emphysema from smoking or people who have a pneumonia, do our oxygen levels actually come into play in determining our respiratory rate, right? So oxygen levels are not the prime stimulus for driving respiratory rate, so right, which is good. So we have plenty of oxygen left in what we exhale, which is why we can do rescue breathing. And if I put an oxygen mask on you, you will not slow down your respiration. So the only people in whom oxygen is an important determinant of respiratory rate are people with lung disease. So this is my grandma holding me when I was little, and she smoked a lot. And so my grandma actually ended up living the ends of her days in a state where oxygen levels were determining her respiratory rate. But in the non-disease state, oxygen levels are not what determines your respiratory rate. It turns out that it's actually carbon dioxide. Carbon dioxide, CO2, is the boss of breathing. So the reason is carbon dioxide is acidic. So as carbon dioxide builds up in the bloodstream, because it's constantly being produced by the mitochondria as a waste product from aerobic cellular respiration, it ends up lowering the pH, because remember, acidic is lower pH. So we can't let that happen, right? Because then our enzymes are going to start to fail. That's really dangerous for the body. So actually, before our oxygen levels even start to fall, it's actually our carbon dioxide concentrations which impact pH that our body has to focus on. And it just so happens that that also ends up making sure that our oxygen levels are kept normal, but that's not the prime driver here. How do we know what the pH is? Well, we have special cells called chemoreceptors, right? Special receptors. We've talked about chemoreceptors before. We talked about them with the cardiovascular system and how if they sense decreased pH due to increased carbon dioxide, they will signal the medulla oblongata to speed up the heart rate and therefore increase cardiac output so that we can get that carbon dioxide out more quickly. So these chemoreceptors not only measure the pH of the blood and signal the medulla in regard to our cardiovascular system, but they are also sending the message to the medulla oblongata that will signal the medulla to have the diaphragm contract and make you breathe, right? So anytime the pH drops too low, we're going to stimulate respiration. The medulla oblongata is going to send that signal down to the diaphragm to contract. And what's kind of crazy to think about is that this is happening 6 to 12 times per minute, right? That's how sensitive these chemoreceptors are to pH, which is an indirect measurement of carbon dioxide levels, right? They're constantly monitoring and then sending signals, every 6 to 10 seconds. So this is also why if you do a breath holding contest, which we normally do if we're physically in class, and you're trying to hold your breath as long as you possibly can. So what's happening here is that more and more carbon dioxide is building up in your bloodstream, and you can't get rid of it. And, it's just simple diffusion, right? So if you're not blowing it out of your lungs, then some more of it's going to end up staying in your bloodstream. So your blood pH is going to get lower and lower and lower. And your medulla is going to really start yelling at you and being like, no, 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 I really need you to breathe. Stop holding your breath, right? And if you want to try to hold your breath just a little bit longer, usually what people do is they will blow out a little bit. And what that does is it gets rid of some of that carbon dioxide. Your pH comes up a little bit, and you can hold your breath for slightly longer. So that's why that little breathing out when you're getting really desperate can buy you a little bit more time, because that's what your medulla is basing it on, right? Is that pH due to carbon dioxide levels. So carbon dioxide levels are what regulate breathing via pH, right? So medulla oblongata signals the diaphragm to contract when pH gets low, and that's communicated to the medulla by the chemoreceptors in the aortic arch, the carotid arteries, and there's also some in the brain. So most of the time when we are healthy and functioning normally, low pH is due to high carbon dioxide. So it's a good proxy measurement. But there are some medical problems that can also cause your blood to get too acidic and to have a low pH. One example of that is diabetic ketoacidosis, which is something that can happen primarily in people who just have type 1 diabetes. So this next video clip that I'm going to ask you to watch is actually a clip of a young male who is in diabetic ketoacidosis. So to give you an idea, normal blood pH is usually between 7.35 and 7.45. Some people with DKA or diabetic ketoacidosis, if they get really ill, their pH can get down as low as 7.2 or even 7.1. It's very rare for people to survive a pH that gets below that, although occasionally it has been documented. Those are really, really acidic, relatively acidic pHs. You might be like, oh, but Dr. Hall, 7 is neutral. Yes, but too low of a pH for human enzymes to function. So what you'll see in this video clip is how much ventilation is happening because the pH is so low. The medulla is sending frequent signals to the diaphragm as well as to those external intercostal muscles to breathe as deeply, rapidly, and forcefully as possible to try to increase the ventilatory rate, increase that air circulation to try to blow away as much carbon dioxide as possible to try to get rid of acid from the system. It's quite something to see. All right, so let's now get down to the nitty-gritty now. Let's talk about gas exchange, right? The whole point of all of this. So carbon dioxide in the bloodstream is not carried around as a free gas, which is a good thing because you can see on the image here, like kind of bubbles in soda, right? If every time then we jumped around a lot, then our blood would get all fizzy. That would be a problem. So we don't carry it as a free gas. We actually, just a reminder, because you haven't heard it enough today, where does it come from, right? It comes from aerobic cellular respiration as a waste product of ATP production. So it's mostly carried as bicarbonate ion or carbonic acid in the bloodstream. So this is the chemical reaction that occurs. So carbon dioxide combines with water in the bloodstream to form carbonic acid, which then very quickly moves into bicarbonate ion, and that is the primary way that we carry it in the bloodstream. In fact, one of the tests we often do on blood tests is to look at people's bicarb levels. So this is something that we can measure and see quite easily. When it's produced in the cells, the reaction is going to move this way. And then when we get to the lungs, we're going to move it back this way into a free gas so that it can move out into the air and be exhaled. We need to do this really, really quickly. And anytime you need a chemical reaction to happen really quickly, you need an enzyme. In this case, the enzyme is called carbonic anhydrase, and that's what allows us to do this chemical reaction very quickly in both directions. Okay, so it allows us very quickly at the cellular level down in the tissues in the capillary bed to move to this in the bloodstream, and then when we get to the lungs, we can do this so we can blow away the carbon dioxide as a free gas. Now, oxygen is also not carried as a free gas, and you might recall that it's attached to molecules of hemoglobin, right, which are the pigment that's in red blood cells that's combined of 4 heme molecules and 4 globin molecules, hence hemoglobin. You'll recall that heme molecules require iron to be produced. So the cool thing about hemoglobin is that you don't need to have an enzyme. It's just super cool that way. It can just grab onto oxygen and hold on to it, and then it can also release it very easily and very quickly. Super awesome. So when we get out into the tissues, so remember, as blood comes, oxygenated blood comes from the heart, out the aorta, all through the arterial system, and is eventually going to get into the capillary bed. That blood is oxygenated, it's high in oxygen. So you'll recall that in the capillary bed, oxygen is going to move down its concentration gradient from an area of high concentration in the bloodstream to low concentration in the body tissues. Simple diffusion. Right? And then this blood is low in carbon dioxide, so the concentration of carbon dioxide is higher in the tissues where it's being produced, so carbon dioxide moves into the bloodstream in the capillary bed. So that's why that blood is returning to the heart. Now to the right side of the heart is going to be low in oxygen and high in carbon dioxide. So blood then is going to come into the right side of the heart, right atrium, right ventricle, out the pulmonary trunk, into the pulmonary arteries, which you'll recall we color blue. They're arteries because they're carrying blood away from their heart, but they're blue because they're carrying deoxygenated blood. So blood is going to go into smaller and smaller pulmonary arteries, and then ultimately a pulmonary arteriole, and then to a capillary bed in the lungs that is surrounding the alveolus. This is the alveolus here. So we have this network of capillaries surrounding the alveolus. Here is where the carbon dioxide is going to leave the bloodstream, move into the inside of the alveolus. Oxygen is going to move from the air inside the alveolus into the blood in the capillaries, and then the blood will continue to move out through a pulmonary venule, which will then drain into pulmonary veins, and then eventually into the left atrium, left ventricle, out the aorta, because this is now blood that's been oxygenated and has gotten rid of its carbon dioxide. All right, and that's the whole thing there. The next slide is a nicer image. Okay, so here's our alveolus, right? These are our air sacs here. So we have air coming in, and then air is going to go out when we breathe. So carbon dioxide is going to move from the arterial, the pulmonary arterial side of the capillary bed out into the air of the alveolus, and oxygen is going to move from the air of the alveolus into the pulmonary capillary, where it's going to bind to the hemoglobin molecules in the red blood cells. So that's what's happening in the lungs, which, as you can see, is kind of opposite of what happens out in the capillary beds in the tissues, right? And it's kind of weird to think about it, but the whole reason that we breathe is to support aerobic cellular respiration. Those are the only things that we need to get oxygen for and get rid of carbon dioxide, because it's just so that we can make that ATP in our mitochondria. It's pretty crazy. So here's a couple of videos on gas exchange, that are also posted in Canvas. I thought they were pretty good. So if you want to review that and some more, there you go. Okay, in-class assignments, just to make sure that people are looking at these PowerPoints or recordings. What is Oliver doing today to support me in his new role as a TA? Well, not much, I tell you. So he is napping on the bath mat in front of a green patterned shower curtain. That is his contribution today. All right, now for our last section of today's lecture, we're going to talk about lung volumes, ways that we measure what's happening during inspiration and expiration, and how we can kind of get we can use these tools to diagnose and evaluate people's lung function and capacity for ventilation. So spirometry is the way we do this. We actually have people breathe out into a tube, and that will displace a certain volume of air. In the olden days, we would have a big drum filled with water, and then a smaller drum filled with air on top of it. And then when you blow additional air into the drum, it displaces some of the water and it floats more, and it's pretty cool. Nowadays, most of them are digital, however, but that's fine. So what do we know? Most of the time when you're just sitting quietly and you're breathing in and you're breathing out and you're breathing in and you're breathing out, right? What we find is that here you breathe in and you breathe out, in and out, right? And it kind of comes and goes and comes and goes, and it's very gentle. This is called tidal volume. This is the volume of air that you breathe in and then breathe out when you're just resting and breathing quietly. This is usually somewhere around 500 cc's or 500 milliliters, right? So it's definitely less than a liter, right, during normal resting breathing. Now, of course, you can, if you want, as you start to, as you get to the point where you do your normal inhalation here, if you want, you can fill your lungs up further. You can breathe all the way up to here. So take a moment and do that. So go ahead and do your normal quiet inhalation, and then see how much more you can breathe in. Right? So you can breathe in actually a lot more volume of air than you typically do. So we call this the inspiratory reserved volume, right? This is the additional volume beyond what you normally breathe in to how much you can breathe in if you need to or if you want to. So I like to think of it as we hold it in reserve for when we want to do a big inhalation. So that kind of makes sense of the term. Similarly, when you finish a normal tidal volume exhalation, you can also blow out more air than that if you want to, right? So go ahead, do a normal tidal volume exhalation, and then see how much more air you can breathe out if you want. Right? Quite a lot, and you can feel all those muscles of forced expiration, right? So that's this volume here. How much more air can you breathe out if you want to? So that's our expiratory reserve volume. And so you see, we have these three different volumes, and if we combine them all together, that is the total amount of air that I could pull into my lungs or push out of my lungs. And so that is called the vital capacity. This is how much air can you move if you're trying your hardest. So a way to get a sense of your vital capacity would be to take in as big breath as you can, and then blow all of it out. Right? So that whole volume of air that you just moved out, that is your vital capacity. And that's really important for us in understanding what somebody's overall lung capacity is. How much air can they move? Now you might have noticed, if you're a detail-oriented person, that our units over here kind of only go down to 2 liters, right? What does that mean? I just tried to empty my lungs completely. Why does that not go to 0? Well, that is our residual volume. So generally you have about 1 to 2 liters worth of air that you cannot exhale. It must always stay in your lungs. And the reason is because we can't allow our alveoli to collapse and completely shut. We always have to have them be a little bit tiny full of air because these are thin little membranes that are moist because they're part of our inner guts of our body, right? So what happens, for example, if you have a Ziploc baggie and there's just a little bit of water inside, right? And if you let it close all the way, now those two sides of the bag are stuck to each other, right? So what can happen if the alveoli collapse fully is then they're stuck and they can't reopen. It's not like you can go in there and kind of peel them off of each other. So we never want to completely empty all the air out, and our pleura, as you'll remember, through that mechanical coupling with the thoracic wall or the thoracic cage, are kind of holding the lungs always open at least a little bit. So this amount of residual air that we can't exhale is called the residual volume. It's what's always in there to make sure our lungs don't completely collapse. I tend to think of it like, there's that peanut butter that you just can't get out of the jar, right? No matter how hard you try, you just can't get it all out. That's what residual volume is for the lungs. Okay, so in summary for lung volumes, and you're going to play around with this in this week's lab, tidal volume is your normal in and out, quiet breathing. How much air are you moving during a normal, typical exhalation or expiration? Inspiratory reserve is that extra you can breathe in if you want to. Expiratory reserve volume is that extra you can breathe out if you need to. And vital capacity is the largest volume of air that you can move, right? So from when you take in the biggest breath possible and then blow all the air out. And residual volume is that little bit that you just can't force out, which is a good thing because we have to make sure the alveoli always stay open. So the other things that we've talked about today to review, we talked about mechanics of ventilation, how the active phase of ventilation is inspiration when the diaphragm contracts and moves downward, creating negative pressure in the thoracic cavity that pulls air into the system, and how the parietal and visceral pleura with that small bit of fluid between them create a frictionless surface and also achieve mechanical coupling to keep the outsides of the lungs basically kind of attached to the chest wall. And we have additional muscles that we can recruit for forced inhalation and exhalation. The control of ventilation is done via chemoreceptors in the aortic arch and carotids that are sensing pH. When carbon dioxide levels get high, pH gets low, and the medulla oblongata then receives that information and signals the diaphragm to contract. Happens about, 6 to 12 times every minute. We talked about gas exchange, which is occurring in the alveoli and their pulmonary capillaries. CO2, which is high in this blood that it's coming from the right side of the heart, is going to move out of the capillaries into the alveolus. And oxygen, which is high in the air that we've breathed in, is going to move from the alveolus into the blood in the capillary. Remember that carbon dioxide is carried as bicarbonate ion, and we need an enzyme called carbonic anhydrase to allow that conversion to happen rapidly. And oxygen is lucky, it's carried on hemoglobin, no enzyme is needed. So that's it for respiratory physiology. In our third lecture, we'll be putting all of these things together and talking about some more cases. Have a great day.